desk, so I'm going to try to mitigate that a little bit with this mat, but we'll see how it actually works. Alright, so chapter 2, host microbe interactions. Alright, let's get on into it, shall we? The human body reacts in many different ways to microorganisms. These interactions can be summarized in the following categories: resident microbiota, all surfaces of the human body, including skin, as well as the mucous membranes that surround the inner parts of the mouth, nostrils, and genitals, and gastrointestinal tract, are inhabited by a distinct set of microbial communities, which are specifically adapted to the local physical and chemical environment. Such normal microorganisms, called resident microbiota, perform extremely important roles. Okay. Turn that down a little bit. Okay. Okay. Number 1. Resident microbiota engage all available binding sites on the host cell surfaces. Thus, invaders have diminished possibility of attaching to the host cell surfaces. Very important. So essentially we're talking about, the microorganisms that live in different parts or on different parts of you or other organisms. Alright? So the first important thing they do is they bind to all the little receptors on your cells and your skin and everything. But in a healthy way, right? They cling onto you in a healthy way that stops bad ones from taking that spot. So take up receptor space. Should be good. Many microbes secrete vitamins that are absorbed by the host and serve important nutritional needs. Wow. Nice. They give you vitamins. Microbes also boost a form of generalized immunity against pathogenic microorganisms that often share antigenic constituents with the resident microbiota. We'll go ahead and just say boost immunity. In order to establish themselves in the host, the invading pathogen needs to establish itself and create a reservoir. Resident microbiota offers stiff competition for nutrients and may produce secondary metabolites that will discourage growth of invasive essentially the good ones take up all the food. Take resources. Resource from bad pathogens. So now we're going to go through some important resident microbiota. So on the skin, you have Staphylococcus epidermis. Staph epidermis. And a species of Micrococcus corneobacterium and propionibacterium are the chief constituents of the resident microbiota on skin. Certain yeast-like fungi, such as Candida, Malassezia, and Trichosporin, may also be found on skin surfaces. However, any microorganism normally present in the environment can also be isolated from the skin. These may include Bacillus species and a wide range of fungal spores. But such microorganisms are not considered constituents of the resident microbiota. They are usually referred to as a transient microbiota. Nose and throat. Staphylococcus epidermis again, Corynebacterium, Neisseria, and Haemophilus species are normally present in the nasal cavity and nasopharynx. So let's shoot out do this. Nice. Staphylococcus aureus can be isolated from the nose and external parts of the ears of nearly 50% of apparently healthy adults. The nose is a primary portal of entry for airborne diseases such as tuberculosis, diphtheria, and influenza. Yeah, so Staph aureus, kind of an important one. It can be pathogenic, but most people have it on them normally. If you're just extra weakened, then it may become bad for you. The mouth. The resident microbiota of the mouth include alpha hemolytic streptococci and various species of Corynebacterium, Neisseria, Lactobacillus, and Candida. Also, a number of anaerobic bacteria are often present under the gums, just below the surface of the teeth. Microorganisms generally present in the air, food, and water can be isolated from throat swabs, but they are not considered resident microbiota. So it keeps bringing that up. That's an important point in that, if I were to take a swab, a bacteria swab of someone's skin, I may find any bacteria that's floating around that's landed on someone's skin, but that doesn't mean everything is resident. Like it doesn't grow on them. It's not their resident bacteria. Same with the mouth. I mean, same with a lot of these, but especially things like the nose, skin, and mouth, just because they're they're your outer surfaces, that entry into your body. So they're where you're gonna interact with the most other microorganisms. highest species of Bifidobacterium, Lactobacillus, Bacteroids, Klebsiella, Candida, Proteus, Enterococcus, and Escherichia, especially E. coli, are normally present in the intestine. In contrast, the stomach, due to its high acidity, offers a hostile environment to the microorganisms. A number of bacteria normally present in the intestine tract are also important opportunistic pathogens. Yeah, so you have a lot of bacteria in your intestines. That's where all your poop is, right? So, shouldn't be a big surprise. But what may be surprising is that most people do have E. coli. Well, not even most, but everyone is going to have E. coli inside of them. It's just not the same E. coli as the bad E. coli, right? There's little differences that make them more virulent, more pathogenic than other E. coli. Sorry. That word. So the female reproductive organ, unlike the male reproductive organ, I'm sorry, I just don't know what I can say. Unlike the male reproductive organ, which mostly harbors skin bacteria, the female reproductive organ affords a very different environment. While its high acidity and the presence of antimicrobial substances such as lactoferrin, which is an iron-binding protein, and SIGA discourage the proliferation of microorganisms, the moist and often nutrient-rich conditions and partial to fully anaerobic environment favor the growth of certain microorganisms. Lactobacillus acidophilus is the main resident microorganism in the female reproductive organ of women of childbearing age. Lactobacillus. So you keep hearing me say lactobacillus. Those are bacillus bacteria that typically consume lactose to, grow. So any lactobacillus are things you're gonna find in like milk and cheese. It's the bacteria that's found in those things, and therefore often your guts and all that other stuff. Just a little fun fact. So, female reproductive microorganisms in prepubescent and postmenopausal stages are usually a mix of skin and intestinal microbiota. Other microorganisms that may be isolated from the vagina include species of Streptococcus, Bacteroids, and Candida, and coliform bacteria. I said the word, pardon me. Elderly women may also carry S. aureus. A disturbed female reproductive environment can predispose the host to a wide range of infections caused by common gastrointestinal microorganisms. So yeah, it's very cute, very true. The resident microbiota is very important for the female reproductive tract. If you lose it, you're gonna have a lot of other bad bacteria trying to get in there. Pardon me. So what are some of our host defenses, right? So our first line of defense, and these are lines of defense against, pathogenic microorganisms, bacteria and viruses, the bad stuff. So our first line of defense, the uppermost layer of skin is comprised of a dead, keratinized layer of cells. Thus, the intact skin is impervious to microorganisms and functions as a powerful physiological or physical barrier. Most of the zoonotic diseases that are associated with insect bites result from the penetration of this barrier. Additionally, several soil and waterborne microorganisms can enter the human body through bruised or damaged skin. Additionally, when the natural flow of the body's fluids, such as urine and bile and saliva, becomes blocked due to a stone formation, this may cause a buildup of bacteria that may lead to infections such as urinary tract infection, cholecystitis, and peritonitis. So that's our first line of defense. It's just our body, pretty much, our physical stuff. The second line of defense is the chemical secretions, the chemical stuff. So examples of this is low pH. So low pH generally discourages bacterial proliferation. Therefore, bacterial growth in the stomach with a pH of 1 through 3, and the female reproductive organ with a pH of 4.5 to 5.5 is generally low. So yeah, the chemicals. Certain iron-binding proteins found in the body's fluids discourage the microbial growth by binding with free iron, which is essential for microbial growth. Some of the notable iron-binding proteins, such as lactoferrin, found in breast milk, semen, and the vagina, and transferrin, found in blood plasma. Lysozyme, an enzyme that dissolves bacterial cell walls, is plentiful in tears and saliva. Interesting. In addition, many body secretions such as saliva, vaginal fluids, and breast milk contain large quantities of a secretory immunoglobulin called SIgA or IgA2, which tends to agglutinate and immobilize invading pathogens. Interesting. We got stuff that dissolves cell walls in your eye and your tears. Cool. Okay, so let me have the third line of defense. This is non-specific immunity. Okay, so first we have phagocytosis. Phagocytosis by neutrophils is perhaps the most active frontier in non-specific immunity. Neutrophils patrol the entire body and engulf and destroy non-self particles such as viruses, bacteria, yeasts. Similarly, activated monocytes called macrophages. Oh, wow, got a long one there. Yeah, that's where it's gonna get kind of dense. I'll try to go through that one kind of fast. Such as macrophages may roam the tissues or gather at the site of infection and engulf the invaders. The macrophages in the liver are called Kupffer cells, and dendritic cells, those in the lungs, are called alveolar macrophages, and those in the kidneys, intraglomerular mesangial cells. The engulfing of non-self entities occurs through a process called endocytosis. The engulfed microorganism is contained in a vacuole called a phagolysosome. So, kind of just describe that. So you got the bad thing here, right? Let's do. Oh yeah, bad. Skull and crossbones. It's a bad thing, right? What it's saying is you have phagocytosis. So this is your cell right here. So a macrophage. You see how it is eating? It is eating this, and eventually it's going to put it inside of it itself. At that point, it is called a phagolysosome. And then it's just going to destroy it inside of its own body. It's going to digest it, essentially, its own body, but it's, the cell. The microbial destruction in the phagolysosome may be oxygen independent or dependent. The oxygen independent mechanism involves a lysosome, lactoferrin, and defensins. So those are little things inside of this macrophage, little organelles that will help dissolve it. The oxygen dependent mechanism kills through the production of NADPH oxidase, myeloperoxidase, nitric oxide synthase, nitrogen peroxide, and hydroxyl radicals. Natural killer cells. NK cells are a kind of lymphocyte that is different from T and B lymphocytes that participate in specific immunity. Like macrophages, NK cells do roam all over the body and seek out and destroy non-self entities that have managed to get into the bloodstream. They recognize self from non-self by sensing changes in cell surface proteins and often result from microbial infections. Malignant cells are recognized in the same manner. The process may involve a major histocompatibility complex protein MHC1, which is found on the surface of most nucleated cells. The NK cells have specific receptors that bind MHC1 proteins. If the cell is from the self or the host. If the NK cell is unable to bind with the cell, it is treated as non-self. The NK cells seem to inject a protein called perforin, which causes lysis of the infected cells and tumor cells. The NK cells also contain Fc receptors. Fc receptors on their surfaces. The Fc receptors combined with the infected host cells destroy them directly by injecting perforin. Wow. They got the NK killer cells, the natural killer cells, so they just inject the perforin when they detect the MHC1 protein change. Protein surface protein, perforin. Okay. Complements, a set of interacting serum proteins, are also components of non-specific immunity. I actually know very little about these. Let me write this. Okay, let me write that down. Complements, a set of interacting serum proteins, are also components of non-specific immunity. There are nearly 20 different types of complement components, named C1, C2, C3, C4, and so on. Their mechanism of action may involve a classic pathway, wherein the complements act on the antigen-antibody complex to destroy the pathogen by injecting perforin in the invading cells. The perforin causes holes to form in the microbial cells, resulting in their death. Another pathway, called alternate pathway, can destroy pathogens before a specific immune response. This pathway is initiated in response to certain bacterial molecules, such as lipopolysaccharides or LPS, found in the cell walls of gram-negative bacteria. Complement C3 is cleaved into C3A and C3b, which bind to the LPS, leading to the formation of an enzyme called C3bbb. Sorry. defense. Which is special or specific immunity. Specific immunity is a pretty dense topic. I will go ahead and read through it. I'm not going to be able to take notes on all this stuff, so I'm just going to probably read through it. Specific immunity is characterized by the presence of a memory system through which components of the immune system remember the pathogen and mount a massive attack each time the pathogen enters the body. Before any further discussion, certain related terms merit elucidation. These are cytokines and antigen. Cytokines are glycoproteins produced by numerous cells, and they play an important role in specific as well as non-specific immunity. Cytokines. Glycoproteins. They include interleukins, interferons, and tumor necrosis factor. The function of interleukins includes, but is not limited to, stimulation of T-cell proliferation, production and differentiation of macrophages, and growth and activation of B cells, in addition to an effect of the central nervous and endocrine systems. Properties of interferons include antiviral and anti-proliferative activities and stimulation of T cells and macrophage and NK cells. The TNFs are cytotoxic to tumor cells and have numerous other properties, including mediating inflammation. As stated earlier, the immune system recognizes foreign bodies via its ability to differentiate between self and non-self using MHD proteins. Any foreign body that is recognized as non-self is an antigen. However, in reality, these are mostly glycoproteins and polysaccharide molecules commonly present on the microbial cell surfaces. Antigens have antigen terminants called epitomes, which bind to T-cell receptors or specific antibodies. A number of small organic molecules may not act as antigens by themselves, but they can act as antigens if combined with protein or similar large molecules. Such antigens are called haptens. For example, penicillin by itself is not antigenic, but when it combines with certain serum proteins, it becomes a hapten and triggers a strong immune response in sensitive persons. So, essentially, cytokines are glycoproteins, like little things emitted from cells in the immune system. And they have a bunch of different responses in the immune system, but the main ones that I know of is like they kind of they recruit immune cells. That's kind of like the main thing they do, right? So if your like lungs are being attacked by pneumonia, some of the cells are going to emit cytokines, and those cytokines then go in your blood and call out for help. They call out and try to get immune cells to come. Antigens. Antigens are pretty much just like anything that is seen as foreign in your body. But yeah. That's kind of the easiest way to describe it. It's not always bad, but your body will usually treat them all pretty much the same. They'll send a bunch of these cells to go attack it. There are two lines of specific immunity: humoral and cellular immunity. It must be stated here that immunology is a highly developed branch of science, and readers who want to develop an in-depth understanding of the topic should consult several scholarly books in this field. Humoral immunity is mediated by B cells, B lymphocytes. B cells originate and mature in the bone marrow, but differentiation occurs in the lymphoid tissue. Activation of the B cells can be antigen-specific or T-cell dependent. The antigen presented by the macrophages stimulates T helper cells, which in turn produces numerous cytokines that act on the B cells. So, whenever an antigen, so a foreign thing, is presented by the macrophage, that big thing that eats it, right, comes and brings it, and that stimulates a T helper cell, which is a developing cell, which in turn, that's just the helper cell, sorry, it produces cytokines, which are just these proteins, right? These then act onto B cells, okay, which are these B cells, which are immune cells as well. That then gives them, I'll just read it, the excited B cells develop into memory cells. B cells do memory cells, very important, very important. Which store antigen-related genetic information in plasma cells, which secrete specific antibodies. Antibodies, also called Antibodies, also called immunoglobulins, or Ig, are essentially glycoproteins that are divided into 5 classes, namely IgG, IgM, IgA, IgD, and IgE. So very important. Immunoglobulins. The building block of these immunoglobulins consists of 2 heavy chains linked together by a disulfide bond. Each heavy chain is attached to a shorter light chain via a disulfide bond. Both the heavy and light chains have a larger constant region and a short variable region, which function as the binding sites. You'll see that in figure 2.8 on the next page. A set of 2 light and 2 heavy chains is also called a monomer. The IgG is a monomer and accounts for more than 80% of total serum antibodies. IgG, so IVIG is very common. They neutralize toxins, opsonize pathogenic cells, and activate complements. A rise in the titer of IgG is often associated with a recovery stage of infection. IgG can also cross placenta. The IgM is a pentamer formed by connecting 5 monomers via disulfide bonds in a J chain. So you can see right there. So that is what these immunoglobulins look like up close, all the way up close, very close. So you have the different proteins, and these are the little things that bind them together into this shape. Yep. Cool. Oh gosh. Okay. The IgM is often the first immunoglobulin to appear in the case of infection, and the titer increases as the infection progresses. IgM molecules are very effective agglutinator and account for 5 to 10% of total serum antibodies. IgA can be divided into 2 groups, IgA1 and IgA2. IgA1, a monomer, accounts for about 5% of total serum antibodies. IgA2, a dimer, 2 monomers joined together with a J chain, like this. Also known as secretory IgA, or secretory IgA or SIgA is found on the surface of most mucocutaneous tissues lining external surfaces, such as the mouth, nasal cavity, and the vagina. I promise I can say that word. It trips me up every time. I just don't want to have any issues with YouTube. It is also abundant in breast milk. IgD monomer accounts for less than 1% of total serum antibodies. They are found on B cell surfaces and play a role in antigen recognition. IgE, also a monomer, is mostly associated with allergy. Interesting. It is present in serum in very small quantities, often accounting for less than 5.05% of total serum antibodies. IgE titers rise during allergic episodes. Interesting. So these are the little things floating around, kind of helping identify and start immune reactions. Okay. Cellular immunity. The other kind of specific immunity is mediated by T cells. T cells also originate in bone marrow, differentiate in lymphoid tissues, but mature in the thymus. The plasma membrane surface of T cells has specific receptors called T cell receptors that bind with the antigens. Two specific components, an alpha polypeptide chain and a beta polypeptide chain, are present on the receptor site. Part of the alpha and beta chains extend into the cytoplasm, while some of their portions remain on the membrane surface. The T cells react to antigen fragments attached to the major histocompatibility complex, or the MCH. Molecules. The eventual activation of T cells is prompted by specific molecular signaling from inside the cells. Activated T cells multiply to form activated T cells multiply to form more specialized subsets, including T helpers and T cytotoxic cells. In particular, T helper cells, are known as CD4 plus cells, are activated by antigens presented by MHC2 molecules on the antigen-presenting cells. Antigen-presenting cells, also known as APC. The activated TH cells perform several functions, including promoting T cytotoxic cells and activating macrophages and triggering production of cytokines, interleukin-2, interferon gamma, and tumor necrosis factor. Alpha, by mediating inflammation. TH cells also stimulate antibody production and play a critical role in allergic response. The T cells, also called DD8 plus cells, do the actual indirect destruction of the pathogen cells or host cells containing pathogens such as viruses or other intracellular pathogen. Activation of T cell Activation of T cytotoxic cells may occur when they interact with APC, generally the macrophages or dendritic cells. Activated T cytotoxic cells destroy the target cells either via the perforin pathway or through the CD95 pathway. In the perforin pathway, the binding of T cells to target cells induces cytoplasmic granules to move to the part of cytoplasmic membrane that is in contact with the target cell. The granules then fuse with the plasma membrane and release perforin and granzymes. Interesting. Perforin polymerizes the target cell membrane to produce pores. The granzymes cells via a process called programmed cell death. During the course of the CD95 pathway, with the activated T cytotoxic cells increase expression of Fas ligands, which are a specific protein. Fas ligands interact with the transmembrane Fas protein receptors found on the surface of the target cells. As a result, target cell apoptosis ensues. So essentially, it blows up. Two other subsets of T cells are T suppressor and T memory cells. The T suppressor cells assist in the functioning of T helper cells and are believed to play a role in the host allergic response. I believe that is the last page. Yep. Alright. Well, thanks everybody for going through that chapter with me. Very interesting to learn, about how things are transmitted in our body. How things are